made up of different components, but the animal cells are not made up of any cell wall structure. They do have something which is called as the extracellular matrix, and this extracellular matrix in case of animal cells are glycoproteins in nature. And when it comes to studying the process of immunity or antigen-antibody interactions, you will understand that. Basically, very next topic when you are going to study about membranes, if you remember, I have told you that membranes contain proteins as well, and you have two types of proteins: the transmembrane protein and peripheral proteins. And these peripheral proteins basically could be glycoproteins, or could be lipoproteins, or could be any type of a protein depending upon the function it needs to perform in a given type of a cell. And that's exactly what is the extracellular matrix or extracellular component of a plant, of animal cell, right? To make it a little more clearer to you, I'll take you to the presentation, and we quickly revise what we have done. When I'm talking about the comparison between a eukaryotic and a prokaryotic cell, this table will help you a big way, wherein you have the size of a prokaryote, which is between 1 and 3 micrometers. Eukaryotes are anywhere between 10 to 100 micrometers. In the DNA for a prokaryote is in a closed loop structure. It is a double helical structure because the eukaryotes have DNA in a double helical structure. The DNA found freely floating in the cytoplasm. The DNA has a nuclear membrane. The organelles are the cell parts in case of prokaryotes. They are membrane bound. In case of eukaryotes, chromosomes are 70s, are small, and in case of eukaryotes, they are bigger and are 80s. There are no mitochondria in the prokaryotes, but there are mitochondria present in the eukaryotes. And if they ask you to differentiate between a prokaryotic and a eukaryotic cell with the help of a diagram, this is what you are going to be drawing. You draw karyotic cells, you draw eukaryotic cells, and you will label the different parts of the prokaryotic cell. You will do the different organelles of a eukaryotic cell. The prokaryotic cell looks smaller. You make the prokaryotic cell look smaller. You have the eukaryotic cell looking bigger than the prokaryotic cell. So this is an exam question which comes from PO, wherein you are asked to differentiate on the basis of a diagram. You get that? Understood?
Yeah.
Right? Okay. This is what we've already done. This is an animal cell that you see. This is a plant cell that you see. And all of these parts, this is the cell. This is your membrane here. These are your chloroplasts. This is your vacuole. This is your nucleus. This is the mitochondria. Then you have the plasmic reticulum, and the Golgi apparatus, just as in case of your eukaryotic cell or the animal cell. And I'm sure you know about your animal cell, and I don't need to discuss this with you, right? Yeah. Now, I'll give you a situation wherein if I give a plant tissue comprises of plant cells. Now, if I suspend this particular plant cell in water, what will happen?
It starts to separate. It starts to increase in size because the water will go through the wall.
And what's going to happen? What is that condition called for a plant cell?
I can't remember.
It's called, the plant cell becomes turgid, right? Why does it become turgid? Because the water begins to move into this vacuole, and then the vacuole becomes bigger. It swells, because there's more of water in it. And as it swells, this hits the cell membrane, and eventually the cell membrane will hit the cell wall, and the plant cell looks more turgid. But if that's the situation, and I suspend this very plant cell in a solution of glucose or sucrose, what's going to happen?
The cell.
The plant cell. Earlier,
it will get smaller, maybe.
Yeah, that's right. Why do you think it's going to get smaller?
Because in the plant cell will go out of the glucose.
Why?
It's searching for the glucose for energy. It needs glucose as a form of energy. I'm not sure.
Okay. Let me to what you just told me. You said that if I suspend a plant cell in water, water will move from the beaker in the plant cell, right? Why did you say that?
Because it's allowed through the membrane. I mean, membrane, through the cell wall.
Okay, so through the cell membrane into the vacuole. But why? Why does water move into the cell?
It's required by the cell. Because of the concentration is lower in the cell than it is outside.
Correct. Concentration of what?
Yeah, of water is lower inside the cell, so the water moves from the higher pressure from outside through the cell into it, and then when you put the glucose on the outside, the water in the cell is at a higher concentration than that of out the cell, outside of the cell. So the glucose, so the water in the plant cell moves out, so it becomes level.
That's great. So I need to push you to remember things, huh?
Yeah.
Okay. At least you're thinking. I'm happy that you think. Right. So what I was explaining to you when we're talking about the extracellular components of a plant cell or an animal cell, and this is exactly what we were just discussing. Flaccidity and turgidity. Water moves into the plant cell, it is called turgid. When the water moves out of the plant cell, it becomes flaccid, and that causes wilting of the plant to take place. You must have observed that when a plant is not watered for a very long period of time, I'm sure that this is it, right? And that wilting happens because of movement of water from inside the cell to the medium outside, right? That's the reason why you're looking at turgidity and flaccidity, and these two terminologies are more associated with plant cells than animal cells. What do you think?
I think it's because water goes out of the plant cell. Because the plants don't have a layer like we have skin, so the water evaporates easily. Where animal cells, they have a skin and means to preserve the water better. We do, water does evaporate off our skin, but less than, it's because the plants have a cell wall, and we don't. It's because the plants have a limited capacity to hold the water, and when that capacity is full, the water comes out. But we still have that capacity, but we don't have a cell wall. Our cells can expand and stretch better than the plant cell. I think.
Okay. No, you correct explanation that this term is more associated with plant cells because they have a cell wall, and we have a cell wall.
Yeah.
So this which I have given you earlier with a plant cell, if I tell you now that I've given you a animal cell, not a plant cell, and the animal cell we put it in water, what do you think is going to happen?
Animal water. The cell will increase in size.
And eventually?
Nothing else will happen.
Nothing else will happen. Then, does the cell help or prevent the plant cell from bursting when suspended in water? Because the cell is increasing in its size, right?
Yeah, but it stopped to a point, won't it?
It would, but the cell is providing a support to it so that it does not burst. But that support is not present in animal cell, and that is the reason why we are looking at a concept called plasmolysis in animal cells. You've heard of the word hemolysis? Hemolysis? Hemolysis?
No.
Hemolysis is the bursting of RBCs. Okay? Like meaning breaking, and heme comes from the word blood, right? So hemolysis is the breaking of RBCs. Now, whenever a little salty, right? And it has a particular concentration of. Hello?
Hello?
Yeah, are you okay? Is everything okay?
Yeah. It's fine.
So there is a particular concentration of NaCl within our blood. Okay, we have sodium ions and chloride ions, and that's the reason why your blood is a little salty to taste. Now, if you perform an experiment wherein you vary the concentrations of NaCl so that they don't really suit the concentration of the NaCl in human body, your RBCs will begin to lyse. The reason being that there is a movement of water into the cell, as a result of which the cells burst. Okay, so that is known as hemolysis. Okay, so animal cells, because they don't have a cell wall to protect them, there is every likelihood of cell bursting to take place, though it is not a very common phenomenon, because in the human body there is a mechanism which is called as homeostasis, which controls all these factors like temperature, pH, water content, so that all of them are imbalanced, which would cause harm to the cells in our body. Is that clear with you?
Yeah.
Okay. Alright. So, your extracellular components could be in the form of glycoproteins, which are cell receptors in our body, or they could be the bone matrix, which is a network of calcium-based compounds that give the structure and strength to the bone, or it could be glycoproteins, which are not receptors, but they help in addition of cells and communication of cells and transfer of information from one to another cell, or it could be the glomerulus that is present as a part of the kidney, which helps in ultrafiltration of the blood, and this glomerulus has a very strong membrane, so the blood can be pushed through it. Now this we are going to understand when we come to the topic of human health and physiology, right? So all we need to remember now is animal cells don't have a cell wall, but they definitely have extracellular components, which could be in the form of glycoproteins, which could be cell receptors, or they could be glycoproteins which help in adhesion of cells to one another, or they could be calcium-based components of the bone, or they could be the glomerulus of the kidneys, which helps in ultrafiltration. So our extracellular component is extra support that the membrane gets from these or calciums or the lipid or something